In the past, we've talked about alkyl halides as a functional group, and that led us into a discussion about substitution and elimination reactions. Now we're going to move on to another functional group, the alcohols. Alcohols, basic structure is an R group with an OH. That R group signifies any sort of carbon chain. The functionality of the hydroxide is sort of similar to that of an alkyl halide in that we've created this polar bond between a carbon and an oxygen, the alpha carbon and the oxygen of the alcohol. Alcohols can be methyl alcohols, primary, secondary, tertiary alcohols, and in a lot of ways act a little bit like water, right? We have this OH group that can accept protons as a weak base. It can donate protons as a weak acid. Alcohols can hydrogen bond like water, and in many cases react in similar ways to water. Now, in this video, we're going to talk about one of the more popular and abundant alcohols, and that's ethanol. Here's ethanol down at the bottom of this slide. Again, we have our ethyl group attached to an OH. This is the alpha carbon directly attached to the oxygen. Now, what I'm going to go over here is how ethanol is made through fermentation. You may have heard of fermentation as a biological process, and we're going to talk about it a little bit and how it generates ethanol, whether that ethanol is to be turned into liquor or through distillation or just made into beer or wine or sake or whatever. This is the process in which humanity has made ethanol for thousands of years. So, if you've ever taken any biology, you remember that there's a process called glycolysis that's used in order to break down our glucose fuel in our blood. Glycolysis takes a 6-membered carbon ring of glucose, breaks it into 2 3-carbon pieces called pyruvate, and oxidizes it like crazy, right? We've got a lot of carbon-oxygen bonds here, which is the hallmark of oxidation when we think about organic chemistry. Now, in that process, we really don't get a whole lot of energy out of it. For each glucose that's converted to 2 pyruvates, we generate directly 2 ATPs. We have some indirect energy of 2 NADHs, and we'll talk about that in a little bit. But really, this process of glycolysis is really pitiful in making ATPs. The good thing about it is it's done in the absence of oxygen. This is an anaerobic process, and so this can continue for a while even without the presence of oxygen, spitting out 2 pitiful ATPs per round. Now, what stops glycolysis from generating ATPs in the absence of oxygen? Why do we need to breathe? Why couldn't we just use glycolysis over and over and over again in order to fuel our ATP needs? Well, it turns out there's some limiting reagents present in this process, and the main limiting reagent in glycolysis is NAD+, right? This is a cofactor that's really necessary for the oxidation of the carbon chain of glucose to form pyruvate. NAD+ becomes reduced to form NADH, and as it becomes reduced, it can oxidize the carbon chain. Now, if you undergo anaerobic respiration long enough, you're going to run out of NAD+, and then you're stuck. Well, it turns out that different organisms have ways to sort of replenish the NAD+, so that glycolysis can continue for just a little longer, even in the absence of oxygen. And organisms do it different ways. Humans, for example, can take the pyruvate and reduce it to form lactic acid. And as pyruvate is reduced to lactic acid, our NADH can be regenerated to NAD+ to continue to fuel glycolysis and get our ATPs. Now, you've heard of lactic acid before. When you do anaerobic respiration or you sprint, you get lactic acid buildup. And there's only a certain amount of lactic acid that can build up before it becomes toxic, and you have to stop exercising or breathe hard to get oxygen in there. It lowers your pH, you get acidotic, you start throwing up. and it becomes a real problem. Now, yeast actually do a different process in order to regenerate this NAD+. They don't typically make lactic acid in the absence of oxygen. They will convert their pyruvate in 2 steps to ethanol. The first step is to cleave or decarboxylate this pyruvate, losing a carbon dioxide, ending up with a 2-carbon acetaldehyde molecule. And then the second step is where the yeast reduces the acetaldehyde from an aldehyde functional group to an alcohol functional group. Here, this is a more oxidized molecule because it has 2 carbon-oxygen bonds. Ethanol is less oxidized because it only has 1 carbon-oxygen bond. And since this carbon chain is being reduced, it means we can oxidize NADH back to NAD+ to replenish the NAD+ stores that can drive glycolysis, making more ATPs. So, how is this done in practice? If we want ethanol as a product, well, we take some yeast, we give it a carbon source. Oftentimes in the United States, that's corn, because we've got a lot of corn. We allow that yeast to metabolize the corn, and we do it in a sealed container in the absence of oxygen, so that in order for that yeast to continue to grow and reproduce, it will need some ATPs. As it makes its pyruvate and exhausts its NAD+, it will convert that pyruvate to ethanol, replenishing the NAD+ to go a little further. Now, just like us with lactic acid, yeast can only make a certain amount of ethanol before it becomes toxic to the population. I mean, in the laboratory, we use ethanol often to wipe down lab benches in order to decontaminate them. So most yeast can only withstand about 5% or around of ethanol before they stop metabolizing altogether. You'll notice that the other product of this fermentation reaction is carbon dioxide. So these are the bubbles we get in our beer or champagne or whatever from carbon dioxide. Okay. Different alcoholic beverages are made from different sugar sources, right? If we want beer, we have wheat beers or barley beers. If we want wine, we use grapes as our glucose source. Or if we want sake, we can use rice, or certain vodkas have potatoes as our glucose sources. And then the yeast will work on that sugar source to make a certain low percentage of ethanol, which we either clean up and drink directly, or we can purify it out through distillation. Since ethanol has a lower boiling point than water, we can distill out the ethanol and get something more concentrated, some more concentrated liquor. So, this has happened in the past accidentally, right? If you get a sugar source contaminated with yeast, if it's sealed up and the absence of oxygen, you'll start to do fermentation and generate alcohol, ethanol in there. I remember finding a juice box that apparently had been contaminated with some yeast, and it looked like it was about to explode because it had made a bunch of carbon dioxide and built up pressure. And when I opened it up, it smelled like wine, smelled like ethanol.